In this lecture, we'll be covering fermentation and gluconeogenesis. And on the title slide, I've shown a couple applications, I guess you could say, of fermentation that we'll be talking about, depending on whether you're a microorganism like a yeast or an animal like we are. Okay, so last time we covered glycolysis and certain feeder pathways into glycolysis. What we're going to be talking about in this lecture comes from 14.3, which is fermentation. I'll mention a little bit about biofuels, which really isn't covered in the text, just very briefly. And then section 14.4 covers gluconeogenesis, which is kind of like glycolysis in reverse. Sort of. There's a few workarounds that we'll discuss. And then we'll also cover at the end of this lecture the pentose phosphate pathway, which is sort of like using glucose as a building block. That'll be just very brief. Okay, so to remind you the fates of pyruvate. So in glycolysis, we have glucose going to pyruvate. Okay, and that's through the 10 reactions of glycolysis. Okay, from there, pyruvate has a number of different fates depending on whether or not oxygen is available. So if there is plenty of oxygen available and there's an energy need, pyruvate will go get broken down further into acetyl-CoA, and then that gets fed in the citric acid cycle to make carbon dioxide and water. Okay, and then that generates electrons for the respiratory chain. Okay, that's under aerobic conditions. When there isn't oxygen around, the fates of pyruvate can be different depending on what organism you're talking about. In things like animals like us, during those anaerobic or hypoxic conditions, pyruvate will be converted into lactate or lactic acid. This in microorganisms like yeast, this process, a similar process when there isn't oxygen, pyruvate gets converted into ethanol, and that's what's known as fermentation. Okay, the process of pyruvate going to lactate in humans, you can also call that fermentation as well. So remember, think back to glycolysis, and in step number 6 of glycolysis, glycerolaldehyde 3-phosphate is converted into 1,3-bisphosphoglycerate. And this was done by an enzyme called glycerolaldehyde 3-phosphate dehydrogenase. Right? And if you remember, that enzyme is using NAD cofactor and converting it into NADH. That NADH then gets fed into the respiratory chain under aerobic conditions. And we'll talk more about this in chapter 19. But that NADH is then the main source of electrons for the respiratory chain. Okay, so then NADH donates its electrons through complex 1, and then there's a big shuttle from that to complex 3 and complex 4, and that's what drives this. The electron transfer is what drives the protons being pumped, and then the proton gradient is what drives the formation of ATP. Okay, so that's just a little preview of what we're going to be covering in chapter 19. But that makes this reaction, step 6 in glycolysis, coupled with aerobic metabolism in that respiratory chain. Okay, when no oxygen is present, so this electron transfer requires there to be oxygen as the ultimate electron acceptor. And when no oxygen is present, this electron transport chain stops. Okay. That's all well and good, but what happens to glycolysis? If NAD keeps getting turned into NADH by glycolysis, pretty soon the concentration of NADH, and you can't recycle it back to NAD if there's no oxygen, pretty soon the concentration of NADH will get really high, and the concentration of the NAD will get lower, right? And if you remember that the NAD is a substrate for this enzyme, okay, and if you're lowering that concentration of a substrate, pretty soon that's going to change the delta G of this reaction, and this reaction won't go forward anymore. It'll start going, reverse and actually be in equilibrium, which would not be a very good thing. That would stop glycolysis. So the cell needs another way of recycling NADH when this happens, when there's no oxygen. Okay. And that's where this term fermentation comes into play. Okay. Basically, in anaerobic conditions, you're running glycolysis, you need to keep that central pathway going. So the generation of energy, or ATP, is going without consuming oxygen or a net consumption of NAD+. Okay. When you go through glycolysis to pyruvate, right, that gives you pyruvate. Then pyruvate, when there's no oxygen, needs to be converted into another product. Okay. And that conversion of pyruvate to another product is what's going to regenerate the NAD+ for further glycolysis. Right. The process, as we'll see in the next slide, is used to produce a number of different food items and also chemicals. Okay. So there's this. You don't need to memorize any of these organisms, but just be aware that there's different fates for the pyruvate or pyruvic acid, depending on what organism you're looking at. Okay, but all of them are doing this for the same reason. They're converting during glycolysis. You're making NADH. When there's no oxygen around, that NADH has to be recycled back to NAD+ to keep glycolysis going. Okay, so there's things like propionic acid in Swiss cheese, lactic acid, very similar to animals, but some microorganisms also produce lactic acid, and examples of those are the microorganisms in cheddar cheese or yogurt or soy sauce. Yeast, I think that's probably the most well-known application of fermentation, is to produce ethanol. Okay, things like wine and beer. There's also microorganisms that can produce acetone and isopropanol, very what we would consider kind of harsh chemicals, but that's what they're converting pyruvate into. And then finally, things can produce acetic acid as well. All right. So animals, they undergo lactic acid fermentation. Okay. So the pyruvate is reduced to lactate, which is a reversible process. Okay, and this usually you can think of happening during strenuous exercise. Okay, lactic acid or lactate builds up in the muscle, okay, and that really kind of puts a limit onto the time that you can do like an all-out maximum effort. It's generally about a minute is what they say. Okay. The acidification of muscle prevents that continuous strenuous work from continuing longer than that. All right. How we get around this, or how we can recover from this, is that lactate is transported from the muscle to the liver and converted back to glucose there. Okay, that does require some recovery time. During that time, high amounts of oxygen is consumed to fuel gluconeogenesis, and that is what restores muscle glycogen stores. And that's called the Cori cycle, which we'll talk about here in a little bit. Okay. This is just from my Strava, kind of like a real-world example of this. It's on a segment it's a little bit less than a minute, 50 seconds, 50 some seconds, 50 seconds, there it is. But it kind of illustrates this idea of about a minute is about as long as you can go all out for. So in this time I was averaging 600 watts for this time, and by the end it really starts to drop, okay, because you simply can't keep going because you're getting too much lactic acid buildup. Okay, so what are the biochemical steps of fermentation in animals? Okay, so pyruvate is going to be converted to lactic acid or lactate. Okay. And this is done by an enzyme called lactate dehydrogenase. Okay, very simple, right? Lactate, it's telling you that it's an enzyme that can oxidize or reduce lactate. Okay, it's really an enzyme that's probably named for the reverse step. But in this case, in fermentation, we're talking about it going in the opposite direction. So what's happening, and normally a dehydrogenase, you would take that substrate and a oxidized cofactor, and you would oxidize the substrate and reduce that cofactor, right? So that'd be the reverse reaction. But in this case, the biological function is the opposite direction. And so what the enzyme's really doing is converting pyruvate to lactate, and you see that they're very similar molecules. They're both 3 carbons. Okay, pyruvate has that carbonyl group at carbon 2. Lactate has an alcohol, so it's being reduced. And NADH, the reduced cofactor, is being oxidized to NAD+. And the delta G for this reaction is very, very negative. Okay, so the equilibrium lies far to the right. Okay. This is a redox reaction. Okay, so what's going on? If we look, why is this occurring? Well, in glycolysis, glucose going to pyruvate, you're creating NADH. If you're not using that via aerobic metabolism or aerobic, the respiratory chain, you need a way to convert that. So pyruvate going to lactate, that is recycling your NADH into NAD+. Okay, and so this is just another way of looking at it like that. Okay, what you're doing here is recycling your NAD. Well, you might think, okay, so if you keep doing this, right, that's not necessarily good, because you're going to get this huge buildup of lactic acid, and you're going to have no glucose left, and that's where this Cori cycle comes into play. So in the muscles, that's where this will be occurring, this fermentation, right? Your glycogen and ultimately glucose is being converted to lactate. Under those anaerobic conditions. Okay, you need a way to recycle that, and so your bloodstream will carry the lactate to the liver. In the liver, you convert lactate back into glucose, and then the glucose then flows back into the muscles where it's needed. Okay, so that, again, that's the Cori cycle. In the liver, it uses ATP used in the synthesis of glucose. Okay, so that sort of puts a limit onto how long you can do this without having another way of gaining energy. Okay. So that's animals. In yeast, this occurs via a different product, and that's ethanol. And in yeast, this is actually a 2-step process instead of a 1-step process. Okay, so when I say yeast, we're really talking about the Saccharomyces species of yeast. Okay. And again, this is a 2-step reduction of pyruvate to ethanol. It's an irreversible process. As humans, we don't have one of the enzymes, the first enzyme in this process, pyruvate decarboxylase. We do, however, have the second enzyme, which is alcohol dehydrogenase, which actually allows us to consume ethanol. All right, in the first step of this reaction, carbon dioxide is produced. Okay, and that's what gives carbonation, or beer carbonation. It's also what is responsible for dough rising when you're baking bread. Okay. Okay, both steps require cofactors. Pyruvate decarboxylate in the first step requires a magnesium ion as a cofactor, as well as something called thiamine pyrophosphate, that's abbreviated as TPP, as another cofactor. And we'll show what that looks like here in a second. Okay, the second step, alcohol dehydrogenase, it's a dehydrogenase, so it uses NADH. So that first step here, pyruvate is converted into acetaldehyde. The acetaldehyde is a pyruvate's a 3-carbon molecule. It loses a CO2, and then acetaldehyde's a 2-carbon molecule that's left from that. Okay, so pyruvate decarboxylase, that enzyme name tells you what it's doing. It's taking pyruvate and it's taking off a CO2. Okay, so CO2 is removed by pyruvate decarboxylase. That gives you acetaldehyde. Acetaldehyde then is converted into ethanol by alcohol dehydrogenase, and that's what recycles the NADH back to NAD+. So this is ethanol fermentation is allowing glycolysis to continue. So you have glycolysis here producing ATP, and then it's converting NAD to NADH. Then you can take pyruvate to acetaldehyde to ethanol, and that recycles your NADH back to NAD. So that allows you to keep glycolysis occurring and keep ATP production going. Okay, so biofuels actually utilize this process of fermentation in microorganisms to produce things like ethanol or other hydrocarbons that can be used as fuels. Okay. Feedstocks from plant material, generally those are plant material that contain, cellulose, or sometimes algae are used for this. Okay, and that's where they're considered renewable, because these feedstocks of plant material or algae can grow with minimal input of outside energy. Okay, so they really require, primarily sunlight to grow these things. Okay. The microorganisms can be engineered to maximize the production of your target, or produce even some non-native compounds. So if I'd ask you a question about biofuels, this is probably the most important information to remember, just the process of getting a biofuel. Okay, so the first step here is you have to harvest what's known as biomass. So whatever your feed material is, whether it's plant-based or algae-based, you have to harvest that. Okay, and then that biomass has to be processed so that the cellulose, the molecule that you're trying to break down in the fermentation process is exposed to the enzymes that will be in the next step. Okay, so in this case, in this figure, it says biomass is cut into shreds and pre-treated with heat and chemicals. Okay. The next step is enzymes acting on, in this example, it's cellulose. So enzymes are chewing up the cellulose and breaking it into individual sugars. Okay, those individual sugars are then fed in the next step to microbes to ferment this into, in this case, it's ethanol. Okay, so this is step 4 is the fermentation process, making your target molecule, using fermentation to make your target molecule. Okay, and then in the last step, okay, that molecule that you've made, you generally are going to have to distill it and purify it before it can be used industrially. There's some novel ways to break down cellulose, and this is an example of one. You're not going to be responsible for this on an exam, but you can use things like nanoparticles and then put immobilize enzymes on these nanoparticles that can first break down the cellulose, and then the nanoparticles magnetic, so you can remove the nanoparticles from the tank fairly easily and recycle them and reuse them. This figure shows the fuel ethanol production. Okay, and this is a little while ago, from 2012, but you get the kind of the idea here. Each country, how much they produce in billions of liters, and then kind of an example of what their feed stock is. So in the U.S., it's generally corn to make ethanol. Brazil here, I think that's sugarcane. So they're in Indonesia, I don't know if those are beets, something like that. So they're different places, different parts of the world can use different crops. And one thing I will say about this is that as more crops are used and crop space is used for producing fuels, it does tend to drive up the price of food as well, because there's less acreage available to produce food. So that's one of the unintended consequences, I guess you could say, of biofuels. Another problem with specifically ethanol used as a fuel is that when you use ethanol or add ethanol to gasoline, you create more ozone from the burning of that, especially when the temperature's over 90 degrees Fahrenheit. Okay, so that's just one of those unintended consequences, again, of trying to use something that's more a greener fuel, a more renewable fuel, but there are drawbacks to it as well. Okay, and then this table is kind of interesting. It just shows you from 2004 to 2013 how much the production of ethanol increased, especially in the United States. Okay, it went up quite a bit during that time. I'm not, off the top of my head, I'm not sure if it has continued to go up or if it's sort of leveled off. Okay. Another aspect of biofuels, if you want ethanol, that's fairly easy to do, because there's yeast that just naturally make ethanol. But another thing you can do is engineer microorganisms to produce either more of the thing that they naturally make, or you can also add in genes to them and block the genes that they have, some of the genes they have, and add others in to make other specific fuels. So this is an example of that case. So we have, and you don't have to memorize this for an exam. It's just kind of a cool example, I think. So you have glucose going to pyruvate and glycolysis. Okay, that's in every cell on the planet pretty much. Okay, then pyruvate going to acetyl-CoA. Nothing special about that, but what they've done in this microorganism is they've blocked the ability to create lactate. They've blocked the ability to create other things like succinate and formate. So all of these other branches off of these pathways they've blocked. So that pushes everything into what is this molecule down here. Okay, so in these genes, they've also added to this microorganism, the ones that are in italic, and so they've created a way to push all the glucose that goes through here and convert it into butanol. And then the butanol can be used as a fuel, right? And they've also created a balance here. If you look at the NADHs that are produced, there are 4 NADHs being produced, going through glycolysis and then pyruvate dehydrogenase. And then they use 4 NADHs in this pathway to get to butanol. So that is also in balance. If you're interested in renewable energies or biofuels, engineered microorganisms, there's a place called National Renewable Energy Lab, NREL, out in Golden, that you should look into getting possibly getting an internship or after you graduate a career if you want to work in that field. It's a pretty big center in the country for that. Okay, moving on to gluconeogenesis. Okay. In gluconeogenesis, what is going on here is that you have different precursors being fed into the end of glycolysis, and you're running glycolysis in reverse to get out carbohydrates. Okay. And so this is a figure from the book just showing, the different ways you can feed into this and work back through gluconeogenesis to get to your carbohydrates. Okay. Here's a little bit more, a figure that's has some more detail to it. Okay, so glycolysis is shown on the left in red, right? The 10 steps of that going from glucose to pyruvate. Okay. Glycolysis occurs mainly in muscle and the brain. Okay. Gluconeogenesis is shown on the right side in blue. Okay, and in gluconeogenesis, the overall is the opposite. You're taking pyruvate and you're converting it to glucose. Okay, gluconeogenesis occurs mainly in the liver. Okay, so these pathways aren't both going at the same time in the same cell. Okay, they in general you can think of it as occurring in different places in the body. For something like I think you can also, if a cell could do both of these, they're not going to be doing both of these pathways at the same time. Okay, it would depend on the conditions that the cell is in, whether it's using glycolysis or gluconeogenesis. Okay, here's just another figure that shows the same information. This is the previous one was from a book. This is not from our book. I include it just to give you another way to look at this. Okay, and here's yet another way of showing this information. Okay, the thing I like about this has quite a bit more information, so you don't necessarily have to read all of this stuff. But what this shows that I like is that it shows you this glycolysis is happening in the cytosol. Okay, but gluconeogenesis, the conversion of pyruvate to oxaloacetate in the first step of gluconeogenesis, that has to occur in the mitochondria. Okay, and then oxaloacetate gets out of the mitochondria by this malate aspartic acid shuttle, converted back into oxaloacetate in the cytosol, and then that's converted to PEP. Okay, so that workaround in gluconeogenesis occurs in the mitochondria, whereas glycolysis, we generally consider glycolysis as occurring all in the cytosol. Okay. These are, again, opposing pathways. They're both thermodynamically favorable, though. Okay, they're operating in the opposite directions, obviously. right, where the end product of one is the starting product of the other. Okay. Reversible reactions, you can think of the reversible reactions in glycolysis. Those are going to be used by both pathways. Okay, the irreversible reactions of glycolysis have to be bypassed in gluconeogenesis, right? If they're irreversible, you can't go just simply run them in reverse, so they have to be bypassed with other reactions. Okay, and other reactions means you have different enzymes, right, which really makes these different pathways. Both are highly thermodynamically favorable, and they're also regulated. These pathways, these specific steps, I should say, they're differentially regulated, and that is they're regulated with different molecules to prevent a not necessarily different molecules, but if one molecule shuts one of these pathways off, it's going to turn the other pathway on. Okay, and that is done to prevent what's known as a feudal cycle. So if they're both of these pathways are going at the same time, glycolysis and gluconeogenesis, what you're just going to be sitting there wasting energy. Right, so the cell doesn't want to do that. It wants to be running one or the other. Okay. If we look at the first step of gluconeogenesis, it's pyruvate to phosphoenol pyruvate, PEP. Okay, this requires two steps, and they're both energy consuming. The first step, pyruvate carboxylase, an enzyme called pyruvate carboxylase, converts pyruvate to oxaloacetate. Carboxylation using this carboxylation is done using a biotin cofactor, which it's similar to the TPP cofactor that we talked about in ethanol fermentation. Okay, but you're doing the opposite. Instead of taking off a CO2, you're putting a CO2 on. Okay. And again, this requires transport into the mitochondria. The second step, your PEP oxaloacetate is converted to PEP, and that's by an enzyme called phosphophenol pyruvate carboxykinase. Okay, and again, this is the enzyme's named a little bit maybe for the reverse reaction because it's phosphophenol pyruvate carboxykinase. Okay. This phosphorylation comes from GTP, which is a little bit unique. We haven't really talked about that being used, at least in glycolysis, right? It's ADP and ATP, but in gluconeogenesis, we're using GTP as a phosphate source. Okay, and this in some organisms this occurs in the mitochondria. Some it's cytosol, so it's really you can think of the first step as being in the mitochondria all the time. This second step it's dependent on the organism. So let's look a little bit closer at this. So the first couple steps of gluconeogenesis, right? So we have pyruvate going to oxaloacetate. That takes ATP. And then oxaloacetate going to PEP. That takes GTP. And here's just an example of this occurring in the mitochondria. In reality, there's a couple different pathways you can use for this. You're not going to be responsible for knowing these different pathways. I'm just putting this out here because it's in the book. Okay, I like this figure a little bit better because it's just a little bit simpler, but maybe it doesn't show this. This pathway, right, so in this pathway you can convert oxaloacetate directly to PEP, and then that can be transferred outside the mitochondria. In this class we'll just focus on this pathway where pyruvate is converted to oxaloacetate. That is then shuttled out to the cytosol to make PEP. Okay, and that's done via malate shuttle. Okay, so it adds another step here. Right, so the first step, pyruvate is being combined with bicarbonate to form oxaloacetate. Right, and that's done by an enzyme called pyruvate carboxylase, or PC. Okay. Pyruvate carboxylase uses biotin, a biotin cofactor. That biotin basically grabs the bicarbonate, the CO2 on bicarbonate, and then moves it and sticks it onto pyruvate. Okay. To do that, it needs energy in the form of ATP. Okay. So the reaction type here is a ligation. You're sticking another molecule onto, you're combining two molecules together, right? So it's ligating two molecules. If we look at the mechanism a little bit closer, at least of the cofactor, so this is the biotin cofactor, okay? And what biotin has is kind of like a long tether to it that allows it to sort of move from side to side. So on one side, it can grab that CO2 group from bicarbonate, and that loading of that CO2 actually is what requires the energy. Okay, then when it has that loaded, it can shift over to the other active site where pyruvate comes in, and then in that enol form, it can attach CO2 to it to form oxaloacetate. Okay. The next step, we have oxaloacetate going to phosphoenol pyruvate, PEP. Right. That's done using an enzyme called PEP carboxykinase and GTP as a cofactor. So the CO2 can be a leaving group here. When this grabs the gamma phosphate off of GTP, okay, what we get out is GDP, CO2. That group leaves. The group that we stuck on there just leaves again, and then we're left with pyruvate that has a phosphate group in the middle, right? That's phosphoenol pyruvate. Okay. This is a lyase reaction if you're interested. Okay, so that's one bypass. Okay, so in that your step 10 of glycolysis is irreversible. So you have to, in gluconeogenesis, you're bypassing that with two steps. So those would be step 1 and step 2 of gluconeogenesis to bypass step 10 of glycolysis. The other reverse reaction workaround that we need are for steps 1 and step 3 of glycolysis. Those are also irreversible. Okay, so let's remind ourselves what those are. So step 3 in glycolysis would be fructose 6-phosphate going to fructose 1,6 bisphosphate. So we need in reverse in gluconeogenesis, fructose 1,6 bisphosphate goes to fructose 6-phosphate. So we need a workaround for that, and that's with an enzyme known as fructose bisphosphatase 1, or FBP. Okay, and the other step 1 of glycolysis would take glucose to make glucose 6-phosphate. So gluconeogenesis, we need to do that the opposite, glucose 6-phosphate to glucose, and that's done by an enzyme called glucose 6-phosphatase. Okay, so these phosphatases are sort of easy to remember because those are going to be the enzymes of gluconeogenesis. Okay, so if we look a little bit closer here, and we're missing a step in here, that the enzymes aren't shown very well, but this would be step 1, step 2, and then step 3. So we have to work around step 3 and step 1. Okay, so in reverse order, reversing step 3 of glycolysis here in gluconeogenesis, we're taking fructose 1,6-bisphosphate to make fructose 6-phosphate, and that's done by fructose 1,6-bisphosphatase. So phosphatase is telling you that it's removing a phosphate group. So you're just lobbing off one of the phosphate groups, the one on carbon 1, and it's just being lost as inorganic phosphate. And you're doing that again, similar thing. Glucose 6-phosphate is being converted to glucose, and you're just losing the phosphate as inorganic phosphate, which you really don't want to do if you don't have to, right? The cell is spending energy to put those on. Okay, to convert glucose to glucose 6-phosphate, you're using an ATP, right? That's one of the priming steps of glycolysis. So if you're just going at this in a cycle, let's say, and that's where the feudal cycle comes in, you're just going to be using a lot of ATP, and you're just wasting that phosphate group. It's not going to anything useful. Okay, so that would waste a lot of energy, and that's why these steps have to be very carefully regulated. Okay, the reaction type for both of these, since we're using water to cut off the phosphate group, it's a hydrolysis. These are both hydrolysis reactions. Right. So as you probably have noticed, gluconeogenesis is expensive. In gluconeogenesis, we have the cost of taking 2 pyruvates to make a glucose. That's cost 4 ATPs, 2 GTPs, which you can think of as being equivalent in energy as to ATP, and also 2 NADHs. Okay, so this is an expensive process, but physiologically it is necessary. Okay, there's things like the brain, nervous system, and red blood cells that generate ATP only from glucose. Okay. So they need a way to get more glucose when glycogen stores are depleted. Okay, so they have to get glucose from somewhere. So things like during starvation or really vigorous exercise, you need a way to get glucose to your brain, or your brain would just shut off. Okay, animals can produce glucose from sugars or proteins. Okay, the sugars, things like pyruvate, lactate, oxaloacetate. Proteins, amino acids, can be converted into citric acid cycle intermediates, which we talk about the citric acid cycle in chapter 16, so we haven't really seen that yet. But those are known as the glucogenic amino acids. Animals can't produce glucose straight from fatty acids. Okay, the product of fatty acid degradation is acetyl-CoA. You can't have a net conversion of acetyl-CoA to oxaloacetate. Okay, but some organisms like plants, yeast, and bacteria can do this. They can produce glucose straight from fatty acids. Right. This is just a little preview of the citric acid cycle. You don't need to know any of this yet, but this shows the different amino acids that can be fed into the citric acid cycle at given points. Okay, and we all know oxaloacetate. If you can convert any of these into oxaloacetate, right, oxaloacetate can then go into the gluconeogenesis. The final thing we'll talk about in this lecture is the pentose phosphate pathway. Okay, and some of these reactions in the pentose phosphate pathway might look a little bit complex, but I want to stress that I won't be testing on the nitty-gritty and the super detailed things about the pentose phosphate pathway. I'll try to keep it to to be more of an overview level, okay? So what you're doing in the pentose phosphate pathway is it's another, when the cell has, a lot of energy on hand, glucose 6-phosphate, it instead of pushing that through glycolysis, it pushes it through the pentose phosphate pathway. Okay, and basically what you're doing is you're converting glucose 6-phosphate in a number of steps to ribose 5-phosphate. Okay. When you're doing that, you're creating reduced cofactors NADPH. Okay, so these enzymes in these steps use NADP+. Okay. And glucose, also I should mention, glucose going to ribose, you're losing a carbon there, coming out as CO2. Okay, ribose 5-phosphate can be used for nucleotides and some other coenzymes like ATP, GTP, those sort of things, DNA, RNA. Okay, so that's kind of what that's good for. So in cells that are replicating, let's say they need to replicate their DNA, you need to do this pentose phosphate pathway to make your nucleotides. The other aspect, the other thing you're creating out of this is that reduced NADPH. And so that can be used for a number of different things. One of them is to convert glutathione, the oxidized glutathione, which is abbreviated here as GSSG. You can convert that back into reduced glutathione. Okay, so you have reduced NADPH, oxidized glutathione. There's a redox reaction. You get out oxidized NADP plus and reduced glutathione. And the glutathione, that molecule looks like this, if you're interested here. And this would be the reduced form because you have this reduced sulfhydryl here. What glutathione can be used for is and we'll see this on a different slide, but it's a good way of preventing oxidative damage in a cell. Okay. The other thing NADPH can be used for is what's known as reductive biosynthesis. So things like fatty acids or sterols, steroids, those are synthesized in part using NADPH for the reductive steps in the synthesis of those. Okay. So pentose phosphate pathway, again, the main products are NADPH and ribose 5-phosphate. Okay, so that's one of the important things to remember about pentose phosphate pathway. Right? NADPH, as I said, is an electron donor in reductive biosynthesis of fatty acids and steroids. It also can be used through glutathione to repair oxidative damage. Okay, and then ribose 5-phosphate is a precursor of nucleotides. So DNA, RNA, and some other coenzymes use the ribose 5-phosphate as a building block. Okay. So this is a little bit more detail now of this pathway. And again, I wouldn't expect you to know all of these enzyme names, but so there's 1, 2, 3, 4 steps total here, and you have an enzyme called glucose 6-phosphate dehydrogenase. All right, and so all these enzymes that we're talking about in glycolysis and gluconeogenesis, now pentose phosphate pathway, they all sort of sound alike. So you're going to have to do it takes some effort to really remember them and recall which pathway they're in, because they all sound, sort of similar. Okay, but glucose 6-phosphate dehydrogenase is an enzyme that uses glucose 6-phosphate, right? That's after step 1 of glycolysis, and it's a dehydrogenase, so it's doing an oxidative reductive reaction. So it's using that it's oxidizing glucose 6-phosphate into this molecule, a lactone. There's a you're taking the alcohol there and converting it into a carbonyl. So you're oxidizing that, and that's what's reducing the NADP to NADPH. Okay. And then you have some other steps, right? So that's one of those oxidative steps. You have another step that just, cuts that ring, and then you lose that CO2 group in the next oxidative step and make another NADPH. Okay, and that's 6-phosphogluconate dehydrogenase. So these two enzymes, I'd probably expect you to be able to recognize those names because they're dehydrogenases, and this is the dehydrogenase is producing the NADPHs in this pathway. These other enzymes, I wouldn't expect you to know the names of. Okay. So it's actually kind of a cool process here. If you look at this, these reactions, you're producing 5-carbon, the ribose 5-phosphate, which is a 5-carbon sugar, but in tissues that need more NADPH than the ribose 5-phosphate. In an example here is liver and also fat tissue. What happens is that the ribose 5-phosphate isn't very useful, so there's a way to recycle it back to the glucose 6-phosphate, and that's done. You can see that here in this scheme, right? You have 2 5-carbon sugars. You can make a 7-carbon and a 3-carbon sugar. One enzyme will do that. And then the 7 and the 3-carbon sugar can go fed back in that same cycle to make a 6 and a 4-carbon, right? And that 6-carbon sugar is glucose 6-phosphate. You can then do another oxidative pentose 5 that goes back into the pentose 5 phosphate pathway to make another 5-carbon ribose 5-phosphate, but more importantly, more NADPH. Okay, so that scheme is shown here. Okay, not going to need to know this on an exam. It's just kind of a cool way that the body can maximize the amount of the NADPH being produced in this pathway when the ribose 5-phosphate isn't the desired target. NADPH regulates partitioning into glycolysis versus the pentose phosphate pathway, and it's done in this step. It can feed back and inhibit the first step of the pentose phosphate pathway. Okay, so that's what's known as a feedback inhibition. So if you have a high concentration of NADPH, that will feed back and inhibit that first enzyme of the pathway. So you have glucose to glucose 6-phosphate. That would be the first step of glycolysis. And then it can either go through glycolysis or the pentose phosphate pathway. When there's lots of NADPH around, that's fed into glycolysis. Okay, when there's no NADPH around or very little, that's when it can go into the pentose phosphate pathway. Okay. And as I mentioned, the NADPH can be used to sort of protect against oxidative damage in the cell. Okay, and this is done again through recycling glutathione. So NADPH can be used to recycle glutathione, to reduce glutathione, which is shown here. Okay, why is that important? This is just another way of seeing that. So with certain things, even as simple as aerobic respiration in the mitochondria creates what are known as super radicals, superoxide radicals, which then can make hydrogen peroxide and hydroxyl free radicals. Okay, those are all very, very damaging to the cell, especially things like DNA. Those can damage in really hurt the cell. So you don't want those around in very high concentrations. So this cells use this molecule, glutathione, to react with these reactive oxygen species. Okay. And by doing that, the glutathione gets oxidized. That's shown in this direction. So you have reduced glutathione here reacting with these reactive oxygen species to block the oxidative damage that they can do. When it does that, it gets oxidized. Okay? And so you need a way to recycle the oxidized glutathione back to reduced glutathione. Okay? And so one of the things that can do that is this NADPH. Okay? NADPH and oxidized glutathione can make NADP plus and reduced glutathione. Okay, so this is one of the uses of NADPH. Right? And in an enzyme that does this is glucose 6-phosphate dehydrogenase. Okay, right? That was that first enzyme in that pentose phosphate pathway that we talked about. Okay, if you have a deficiency in that enzyme, it can be very serious, and it can even be fatal in cases of high oxidative stress. So a person that has a deficiency in that enzyme, that is given certain drugs or exposed to, herbicides or even some foods, that can generate a lot of oxidative stress, and they have no way to counteract that, or their protection against that is greatly diminished, and it can even be fatal if their deficiency in this enzyme is high enough. You see this deficiency continue on because being deficient in this enzyme gives these people a certain resistance to malaria because they have a high oxidative stress environment in their red blood cells. So the malaria parasite really can't live in them. So that's why this has been kind of passed on and continues to be seen in certain populations. Okay, so in summary, in chapter 14, most importantly, I think, is glycolysis. That's the process which cells get energy from converting glucose to pyruvate. Okay. Today we talked about gluconeogenesis, which is sort of doing glycolysis in reverse to get carbohydrates out of the feeder metabolites. And then also fermentation being a way to continue glycolysis when you don't have oxygen around. And then finally, the pentose phosphate pathway, that's a pathway that produces NADPH and ribose 5-phosphate.